Good morning, everybody. It's a real pleasure to be here, and I really do believe that this may be one of those steps moving Ayurveda forward. What I'd like to talk to you about is simply how to begin to do that in clinical trial methodology. What I'm going to do is talk to you about the requirements for a well-controlled Western clinical trial, the requirements of Ayurvedic medicine in that context, and one solution that is possible to move forward. When you talk about Western clinical trial methodology, you talk about a well-controlled trial. In each case, when one does that, you need to think about the population, the endpoints, the control for bias, and the analysis. On the other hand, when you're talking about Ayurvedic medicine, you have to think about a holistic style, which involves all of the things you see there: dietary, lifestyle, various types of therapies, and very much emphasis on individual, ever-changing therapy as you see patients. So when we start talking about this whole business, we talk about well-controlled. So what does well-controlled mean? In this case, it means methotrexate. Methotrexate is the basic therapy for rheumatoid arthritis in the Western world. It's the thing we use right from the start. The doses are the ones you see there, basically going between 15 and 25 or 30 milligrams a week, but on a milligram per meter squared basis. And I think that's important when you talk about other countries than the relatively obese ones in North America. And so when you have smaller people, you better go with milligrams per meter squared. And that made a difference, and should make a difference, as we do our analyses. How can it be given? It can be given orally or subcutaneously. And in general, should be given in only one way if we're talking about a clinical trial, not both, because the bioavailability of the two drugs of the drug is different. Does it work? Well, yes, it does. There's something called the ACR, American College of Rheumatology Combined Indices, 20%, 50%, and 70%. They're a combined index. You look at joints, you look at how patients feel, you look at how they function, you look at an acute phase reactant. And an ACR 20 is the kind of measure where the patient will come to you and say, you know, something's happening. An ACR 50, something is really happening. An ACR 70, wow, I'm feeling like nearly normal. So if you think about that, from the point of view of getting a drug registered, an ACR 20 is sufficient because it tells drug A from drug B. But from a clinical point of view, you'd like to see ACR 50s or 70s. And what you see here is across various trials, where I just looked at the methotrexate arms, you can see that in general, methotrexate gives you a pretty good response by an ACR 20, about 50 or 60%. However, when you get to the ACR 50s, it's only about 40%, which means that 60% of patients don't quite get there. It indicates, in fact, we need to do better. But it does show that methotrexate works. Does it affect radiographic progression? Yes, it does. It slows radiographic progression, although it does not stop it. In our world right now, we do a lot of treatment with biologics, very targeted therapies, very effective, much more effective, in fact, than methotrexate. However, extraordinarily expensive and therefore really unavailable. To give you a feel for this, methotrexate costs about $700 a year. These drugs cost $20,000 a year. So that's the way it is, and that's why, when we're talking about well-controlled trial in the developing world, we probably use methotrexate as the basic comparator. So, knowing what we're going to use as a comparator, the first thing you have to ask is, who is it going to be given to? What is the population? And In rheumatoid arthritis, it is patients like this. And you see here a patient with multiple swollen MCPs and PIPs. That's metacarpal phalangeal and proximal interphalangeal joints. You also see some of the systemic effects. So you see the intrinsic muscle atrophy that is part of the disease. You can define it, and there's a new definition that's come up, which is really very good. It quantifies the diagnosis. But basically what it says is the more small joints involved, that's the top portion, the more likely it is to be disease. High titer rheumatoid factor or anti-citrullonated cyclic protein, CCP, helps make the diagnosis more specific. Longer duration of disease helps a little bit. And elevated acute phase reactants help. You put all those together and you have a definition which is about 92% specific and 93% sensitive. A very good place to start. So we can define, by Western standards, rheumatoid arthritis very well. So if you can find the patients, how do you measure them? Well, in many ways, you could measure survival. And here you see a group of patients who are rheumatoid factor positive and negative, and their survival over years. But you see that to show a real difference takes 4 or 5 years. We're not going to be able to do clinical trials over 4 to 5 years. We must find surrogates that can replace that in much shorter periods of time. And the one that we chose, and which is really quite useful, is called the DAS, Disease Activity Score. And it's very simple. I'm being sarcastic. Look at the bottom. You have to use square roots. You have to use natural logs. It is anything but intuitive. However, when it's used, you can see that you can define remission, low disease, moderate disease, and high disease activity. And you can do both a state measure, how are you now, and a response measure, how are you over time. So this measure, which is fully validated, can be used in rheumatoid arthritis and is a reasonable endpoint. To do it, you have to measure the joints. And these are the joints that are measured. You can see them in purple or magenta here. And there are 28 joints that are examined. Well, you as practitioners know that the place that the disease really hurts early is the feet. So how come the feet aren't involved in the measure? Turns out that we're bad at measuring feet. That is, the feet hurt, but we are very poor at finding out if they're swollen or not because there are lots of fat pads and things of that sort. So we found when measuring both the feet and without the feet that you got a better measure without the feet. So the fact is we do a 28 joint count and it takes some training. I was showing some of you yesterday how to do it. It's not quite as simple as it sounds, but bottom line, you examine for tenderness and swelling, and you add it up to get a total semi-objective measure of joint swelling and tenderness. Very importantly, you have to control for bias, because you want the patient to get better, the patient wants to get better. How she feels about what she's given will result in her feeling significantly better or not. So in some way, you have to control the bias. The way to do that is double blinding, or even triple blinding. Double blinding means that neither the physician nor the patient knows which of the test medications they're getting. Triple blinding means that the statistician is also blinded. Well, when you think about Ayurveda, you think about a whole group of different ways of giving the drugs. And you see all those, you know all of these, but there are, I know I can count, there are 6 of them there. And it means that patients will be given different amounts of these drugs over time depending on how they respond. It also means that you as practicing physicians will know what you're given because there are so many of them. How do you deal with that? You deal with it by making placebos. And here are examples of two of them. We don't try and make placebos for each of the hundreds and thousands of herbs, but we do make placebos of how they're given. And you can see here for Kashyam the characteristic of the drug, of the application, and the characteristics of the placebo. So the first thing you ask is, oh, come on, the patient can tell the difference. Well, in fact, we tested them, and they have no idea. They can't tell the placebo from the real of any of these. So we tested that formally and found that, yes, from the patient's point of view, we can give a placebo. And they will remain blinded. If you want to examine the study, you have to analyze it. So you have to think about carefully how to analyze it beforehand. There's a measure that's called a primary measure on which you decide how many patients are going to be in the trial. And then there are a number of secondary measures which help describe what's going on. And then there are exploratory measures which say, you know, I'd like to look at what's going on here. So, for example, a primary measure could be the DAS. A secondary measure could be the ACRs, or the tender joints, or ESR. By doing this before you start, you can get a good handle on what you can expect and how to do the study well. So we applied those things to this study. This was a pilot study. It is not a definitive study. And we need that definitive study. But as a pilot study, we had a few patients in each of 3 groups done blinded. So you could treat with Ayurveda, any of the applications, and methotrexate placebo. Of course, we had a methotrexate placebo. You could treat with methotrexate and Ayurvedic placebo, since we had 7 placebos. Or you could do both. And we did that over 36 weeks, and we took a big chance. Because we asked, at the end of the trial, which group do you think you're in? And if it was unblinded, and the patients and the doctors knew exactly where they were in, trial's over. Doesn't work, because it isn't blinded. So we took the chance, and this just tells us what we were doing. We sponsored it. But here's what we found. We looked at the allopathic physicians, and they were all wrong when looking at methotrexate placebo. And they were wrong to varying degrees in the other groups. So overall, they were only correct 1/3 of the time. Clearly, they were blinded. You would have expected 1/3, 1/3, 1/3, since there are 3 groups. The Ayurvedic physicians, nearly precisely the same. So they were blinded. The patients, precisely the same. So we were able to tell ex post facto that this was in fact a blinded study, and it was double-blinded. This shows those results. It basically says 1/3 of the time people are incorrect no matter what. So it was possible. And what's very important is it allowed the Ayurvedic physician to change their therapy as they needed to during the trial. Because if they needed to change from one preparation to another, or from one dose within a preparation, they could do that. And it did not unblind the trial. Consequently, we now can actually do trials with Ayurvedic medicine at the standards that are expected in Western medicine. So why is that important? Well, if in fact you didn't want to spread the word about Ayurveda, get credibility for Ayurveda, you don't need to do this. But if you want to send this out to the world, you must meet their criteria. Consequently, this allows it to be done. What actually happened was that we were prepared to put in one of 148 different compounds within those preparations if the physicians wanted it, knowing it would remain blinded. But in fact, they only used 40 of them. Nevertheless, that meant that the Ayurvedic physicians could and did put in their feeling of what needed to be done. And even though they could put them in, if they were in the placebo group, they weren't put in. They were just in the placebo. So it remained double-blinded throughout. There were two people who had to be unblinded, and they were the pharmacists. And so we had to be sure that the pharmacists didn't talk to the patients. And we were able to do that, of course. So what were the patients like? Here they are. They were absolutely, they weren't exactly what you'd expect in the Western world. In the Western world, in general, the age is about 52, 53. So these patients were a little younger. They had very early disease. They had, in most cases, were rheumatoid factor positive, therefore more severe disease. About a third of them had erosions, even though they had very early disease, which is what you expect. And they had high disease activity by the DAS. Remember, greater than 5.1 is high disease activity. So now we have young, early, severe patients with high disease activity. So that's the group of patients you really most want to have. Now I want you to memorize this. In fact, all this does is tells you what we had to go through to get the patients. We started out with a health fair and started out with 250 patients that we started to screen. And about, I'd say, 3/4 of them were excluded during the screen. They had OA, they had some exclusions like they were on prednisone or they were on methotrexate already. So we excluded 3/4 of them right off the top. Then we had about 60 left. By the time they got to randomization, 20 of those dropped out because they either weren't interested or they got into trouble in some other way. They fractured or they used another medicine or of some sort. So they ended up with 43 patients. We wanted 45, we got to 43. And those are the patients in the trial. So you have to realize that one thing about this trial is you can't say that it's generalizable to everybody. If it were generalizable to everybody, we'd have 100, 250 people. Okay? So it's a trial that's aimed at the young, severe, active patient. This is the kind of outcomes that were done. We did blood tests, we did tender and swollen joints, we asked the patient about their pain, we asked how they felt overall, and here are the results. And we need to stop and talk about these just a little bit. This is looking at the change from the beginning to the end of the trial. And you can see that the methotrexate group, that's the, I don't know, pink-orange thing. This one over here seemed to have some response, slightly more than either of the other two groups. Interestingly, more response than the combination as well. But it was not statistical. It's a small group of patients. In this case, you conclude that the DAS28 was not different among the three groups. And if you look at the pain and you look at the global, you see basically the same thing. But note that the blue, the combination, was a little less than either one alone. And I think we need to talk about just a little bit towards the end. And if you look at what the Ayurvedic doctors thought globally, you can see that again, everybody improved, there was no difference among the groups. The other side of efficacy is side effects. And here you see an outline of the number of side effects. One of the things you have to realize is that there were side effects in the Ayurvedic group. Another, that the combination was associated with more side effects than either one alone. So let's look at that in detail. General side effects, I feel lousy, doctor, were more common, although very uncommon, in the combination group. And side effects in the mouth, which are very common with methotrexate, with mouth sores, were actually equally common across all groups, including the Ayurveda ones. And cardiovascular side effects, more common in the combination. None of this is statistically different. We're just looking at numerical differences. Very interestingly, in the combination, there was a clear trend to more side effects in the GI tract than with either group alone, although there was some dyspepsia in both. And finally, the neurological side effects, headaches, I just feel lousy, are classical for methotrexate and were more common there, although again, not statistical. So what did we find? Well, we found, very importantly, that there was no difference in efficacy, perhaps a slight trend to less efficacy in the combination, and there were more side effects with the combination. What does that mean? Well, it may mean something we haven't thought about, which is there could be a negative interaction between the methotrexate and Ayurvedic medicine. From a physiological and pathogenetic point of view, that makes sense to me. Methotrexate affects a certain part of the immune system and suppresses it. One of the tenets in Ayurveda is that it increases the immune system. But it doesn't do it across the board, it probably does it selectively. Consequently, I can easily imagine the two interacting negatively. One decreasing, the other increasing exactly the same area. Consequently, not getting more effect, but getting more side effects. So, if we are going to move forward, we have to be very careful about combining allopathic medicines and Ayurvedic medicines. We can't assume there's no interaction, there could be one. We can say that complex Ayurveda can be done to the highest scientific standards, and that's very important. It can allow the Ayurvedic physician to do what he or she wants to do in taking care of the patient without breaking the blind. In this pilot study, it appears that there's no difference between the standard Western and the Ayurvedic medicines. I think it's very important that we move on. This is a pilot study, it's encouraging. We've got to do the full-scale 210, 220 patient study. One could use all three different therapies, but I think that makes it a 350 patient study. It's pretty difficult. We can do one versus the other, and we can do something very important and unique in that situation. We can get samples of blood, plasma, cells, put them aside, and use them for genetic studies, epigenetic studies, cytokine studies, and actually look at the translational and basic aspects of both medication, both treatments over time to see if we can understand how they work better. So we can get a clinical study that will define what the place of Ayurvedic medicine is versus the standard medicine and understand it better. We need to do other studies to understand the potential negative interaction between Ayurvedic therapy and methotrexate. I wanted to thank the people who really did all the work. We helped design it. Dr. Venkatraman is the project director. She just flew back and forth. She took care of all sorts of things to make this thing actually work. There were a number of people at the trust here who actually carried on the work. And there was an allopathic physician who made the diagnoses from the point of view of Western medicine. We had a couple of very good statisticians. And so I think this was not an effort by me. It was the classic team effort crossing allopathy and Ayurvedic medicine. Thank you very much. Now, are we going to take time for questions or not?
A couple of questions.
Okay, so if there are any questions, please stand up and I'll be glad to try and answer them. Yeah?
I have a question. In my opinion, we have a problem with RCTs and Ayurveda, because Ayurveda is not only herbal medicine. Ayurveda means also dietary advice, a daily routine, and manual therapy, and mental or spiritual components. And this you can blind or double blind.
Actually, we can. The question was, so what we've done is looked at one aspect of Ayurveda. We haven't looked at the oils, we haven't looked at the enemas, we haven't looked at the diet. The diet is pretty hard, but I think that all the others are quite amenable to placebo control. So we were prepared to do placebo control for the oils by having a different person do the oils from the one who's examining them. And we were quite prepared for doing placebo-controlled enemas. So the diet was not so easy. But otherwise, I think it is quite possible to do the full range. And in fact, one of the trials we hope to do in the future is a double-blind trial of inpatient medicine, which frequently uses enemas, etc. So I think it's a great question. It needs to be answered, and we're ready to do that. Yeah.
So you said Ayurvedic treatment and acute result could be valid. That was one of your conclusions. But if you see that one of your slides mentions that the side effects were very much less in only Ayurvedic treatment group. So in terms of risk-benefit analysis, how you will comment on this?
So I'm trying to understand what you said specifically. First thing we need to understand is this is a pilot study. It does not definitively prove anything. We must do the full study to get the power to know that they're equivalent. It's encouraging data. If you look at risk-benefit here, there are lots of ways of doing it, but off the top of one's head, one would say that the risk-benefit is against the combination and equivalent for the other two. Okay? Perhaps trending slightly towards a risk-benefit in favor of Ayurveda, because the efficacy is more or less the same and the toxicity looks a little less. But you need to be very careful. One has to be very critical and conservative here. You need the number of patients so you can get definitive numbers to do this. That's why we're hoping ultimately to do the full phase 3 trial. It's a good question.
Do we have any more questions?
I would like to give only a comment. That was our first observation when we started Ayurveda treatment in our patients. For example, we gave to them aspirin in combination with Ayurveda treatment. Then we saw more bleedings in the stomach compared only with the aspirin group only. And we introduced the aspirin dosage to reduce the side effects. Or the same was with insulin. The insulin was much more effective, and we have to consider that the blood sugar does not drop down too much. Yes, I think it's very important to look for the combination therapy and also for the side effects. I believe that Ayurveda drug have a lot of side effects, whenever many people tell to me they have no side effects.
Yeah, I'm sure they have side effects. Everything has side effects. Just to give you a feel for this, in a standard trial where you look at everything, the average number of side effects in the placebo group are 86%. Okay? So it's not a surprise to me that it does. And I think the fact that we saw this and you saw this in terms of combinations and negative effects mandates that we move forward. And I think that Ram is thinking already of doing animal studies, good dose response pharmacological animal studies looking at the specifics of the interactions. Definitely needed.
I have a student who did a thesis in Assam on diabetes and compared an Ayurvedic formulation she had formulated with metformin. And she had put into the Ayurvedic formulation something that she believed would severely reduce the side effects of metformin, and the combination indeed had vastly reduced side effects from the metformin. So the combo group was actually much better because she'd specifically allowed for something that would reduce metformin. Couldn't you do something that would reduce the methotrexate effect?
So I think here's the principal issue here. We always talk about combinations, or often do, in terms of a negative effect. It can also be a positive effect. And consequently, you can do that. Certainly, as a clinical pharmacologist, I would be more than happy to do that. I think it gets a little complicated because there's so darn many constituents in Ayurveda, but there's nothing that says it only goes one way, it could go the other. You were going to say something further?
Yes. I would like to ask you how you make the definition of placebo. We know that we can double or triple the effect of placebos. And are that real placebos, or what is it? They must have an effect in the body. And then we have the question, are these real placebos?
Okay. So the question is, what's a placebo? A placebo is defined as not having the active tested drug. But you need the placebo trials because the mind is a very powerful thing. And if you use a placebo, on average, in rheumatology, you get a 30% response. And that response is real. Acute phase reactions go down, cytokines go down, but it has nothing to do with the active tested medication. It has to do with the power of the mind. And when I talk to patients, I tell them when I'm doing a placebo-controlled trial, I'm not doing a placebo. I'm testing the power of the mind versus the power of the medication. And so that's why we must have placebo-controlled trials. Because 30-ish% of what is going on is due to our ability to control our bodies. So a placebo is only that which doesn't have the active medication. All right. Thank you very much.